Hello, everybody. World Microbiome celebrations. Please confirm in chat if you can hear me. Wonderful. Good evening to all my friends, colleagues, subscribers, fans, critics of BioTecNika. Welcome to World Microbiome Day celebrations. Please confirm in chat if you can see me and hear me. Okay, fantastic. Welcome to this wonderful webinar on this wonderful weekend. I hope all of you are doing great. Okay, fantastic, fantastic. So today we have gathered together to celebrate World Microbiome Day. I hope all of you are aware what is a microbiome, right? Before we even start the celebrations. Microbiomes are an integral part of our world. And today, we are not just going to recognize them, we are not just going to give them a recognition, but we are going to celebrate their presence. In this world of coronavirus, the entire humanity, is scared, threads from the word virus or a microbiome or a microbe. But it is very important as a bioprofessional for all of you to understand the significance of microbiome and spread awareness that not all microbiomes are rogue like coronavirus. Am I right, guys? And that is the reason today we come together once again to celebrate another grand occasion, World Microbiome Day. Before I get started, I would like to thank the co-organizers, Ketek and Abel, who have enabled us to conduct this mega webinar. And before we get started, I have a small presentation to make where I would like to introduce you all to today's agenda, today's speakers. So if you all want me to get started, quickly type in the comment box, BIO, bring it on. Type in the comment box, BIO, bring it on. Yes? Okay. So let's get started with today's presentation. World Microbiome Day. Today is the day where we wish to change the perception and the perspective of every bioprofessional and every common man about microbiome. Today's day is very special for every bioprofessional. And that is the reason we asked all of you to join us, even though it is a Saturday evening, to celebrate this occasion. I can see a lot of BIO coming in. This is what excites us, energizes us to deliver the max to all of you. So 27th of June, 2020, we are celebrating World Microbiome Day to not just give recognition, but to also celebrate the presence of microbiota. How many of you are aware of Human Microbiome Project, which ended in 2012? I hope many of you must be. So microbiomes, as I said, is an integral part of our life, our body, and of course, all around us. So today we are going to discuss microbiome perceptions and perspectives. Now let us be thankful to microbiomes, a community of bacteria, fungi, and viruses that are found everywhere, whether it is within us, around us, in water bodies, as well as soil. Let us be thankful to the microbiome who contributed to the initial oxygen formation on Earth, due to which today we exist. They are 3 times more on our body than even our individual cells. So it is truly said by somebody that if our body is a country, then microbiome is a state. And we need contribution of every state for the progress of our country. So that means we need the contribution of microbiome, and that means we must take care of our microbiome. So we also should know that microbiomes have been seen to contribute to production of electricity and whatnot. The future of microbiota and microbiome is huge and a lot of scope. These microbiomes have formed a billion-year-long partnership with plants as well as animals as well as humans. So today, I would like to request you to become a microbiome savior. Let us celebrate this day by spreading awareness about microbiota. Let us be the savior of microbiome. So if you all agree with me, I would urge you, request you, if you understand the importance of this topic, then please share the link of this webinar with all your friends, family members, and colleagues. Do not forget to put the hashtag microbiome savior, and don't forget to tag us at @BioTecNika, and of course our co-organizers at @KTech_BFC and at @Able India Bio. By doing this small contribution, you will be able to create awareness across the globe, because each individual who is watching this live broadcast matters. And I know you will do that. So quickly, guys, take a minute, take 10 seconds. It just takes 10 seconds. Go ahead, share it on your social media, so that entire biosciences community can come together on one page and have this valuable conversation with our speakers of the day. And we can, so that we can have a mega grand celebration on this World Microbiome Day. Now, we always keep saying microbes, the wildlife needs saving, right? So even the microbes need saving. I found this nice creative by World Microbiome today, microbiomeday.com. So microbes need saving too, and that is the reason we are conducting this webinar today. Now, it is going to be a mega webinar. Like I said in the my earlier section of my introduction, it is going to be a mega webinar because we have speakers from all across the globe coming together on this mega platform to talk just to you about microbiome. So this is not going to be ordinary webinar. So I would urge you, request you to attend this webinar till the end. That will be really a great help. And do not forget to, yes, share the webinar link. So to start with, today's first speaker would be Dr. P.M. Murli, founder, director, Jananome Private Limited, Bengaluru, and a member of Vision Group on Biotechnology, Government of Karnataka. He has so many achievements that it was so difficult to put it all in one slide. So I would just put a summary here. Rest you can always go ahead on the webpage and read about him. He has several contributions to the biosciences community. And you can see today, even though it is a weekend, he comes to you live to talk to you about this subject, microbiome. Followed by him will be our second speaker, Mr. Manoj Dadlani. He is a CEO of Cosmos ID, Maryland, USA. He is the CEO of this company, which is an industry leading provider of bioinformatics solutions, and it helps in unlocking the microbiome. Prior to that, he has worked in several biosciences organizations, and he has been a co-founder as well as a CEO of Rasa Industries Limited, USA. So he will be with us shortly live. After that, we will be joined by our next speaker, Dr. Anindya Das Gupta. Now, Dr. Anindya Das Gupta represents Unilever. He'll be joining us shortly live, and he is the Global Operations Manager, S&T Beauty Products and Personal Care Research and Development at Unilever, Bengaluru. So he'll be joining us shortly live. But before that, I would like to thank and highlight the special role of Association of Biotech Led Enterprises, short form ABLE, and KTech, Department of IT, BT, NST, Government of Karnataka. These two organizations have worked tirelessly with us for past 1 month to make this webinar, mega webinar, successful. So I would like to thank for the special role which Dr. Bala and Dr. Suresh have played for this webinar. I would just quickly check in if we have Mr. Suresh available so that we can have a discussion with him. I don't see him here right now. No problem. So we'll move ahead. Mr. Narayan Suresh, COO of ABLE, has been a catalyst in organizing this webinar, and of course, Dr. Bala from KTech. So without taking much of your time, I would like to pass on the controls to our first speaker of the day, Dr. P.M. Murali.
Good evening to one and all. It's a pleasure to be part of this distinguished panel on the World Microbiome Day. So it's nice to say happy birthday to your better half. In fact, it's a little more than the better half because there are so many of them inside us who are part of us and who have been helping us all the time. So my topic for today is going to be on the let's say fringes of being the 35,000-foot view on how research is going to be progressing, especially using the human microbiota and what's likely to be the outcomes in the future. The first and foremost thing is we have to understand that there's almost 1 trillion microbes inside us. We have always taken note of the rogue ones, which, as you know, are the ones that cause a disease, diarrhea, and whatnot. But the silent ones are the ones who have actually helped us to metabolize some of the things that we cannot, and thereby contributing to what are called as small molecules. These are chemical signals, basically, which try to communicate something. They talk to each other. And also a lot of these small molecules are known to append the health outcomes, including the way the drug is metabolized, including the way we are eating and progressing and so on. But what's going to happen is that there is an innate ability for these microbes to be used as a diagnostic and therapeutic tool, which is where the world is slowly moving towards it. But what is the most important part is that biology, as usual, needs the support of a number of other allied disciplines as well. And one of the areas that the subsequent speaker and my good friend Manoj is going to be touching upon, he's a world authority. His company is the authority in the world. We are very fortunate to have him also in the discussion today. He will be talking about how does he go about looking at these microbes? How does he really even manage to look at all the things that they put out or secrete or metabolize and throw out? What sort of microbes live? And also what are the good microbes and the bad microbes that we need to have for having a great metabolic health? Now, this is going to need huge, very, very huge amount of data and also very large amount of data crunching that needs to be done. And I have a feeling that very, very slowly, this is going to move towards using several intelligent ways of mining this data, correlating this data, putting together and trying to understand. So there has to be a human behind it, but it could be more AI driven in the coming years, whereby a lot of health outcomes of the people, particularly person to person, meaning that you're talking almost like a personalized nutrition, personalized medicine, personalized way of looking after the metabolic health and so on. So the well-being of a person is slowly going to move towards a very individualistic terrain where a lot of the one-size-fits-all type of strategy is going to slowly go away. But believe me, we are in the very, very early days. I think there are about 7,000-8,000 publications which are of significance in this area. A lot of us do follow this area. It's so exciting because we are trying to learn about the better half, about our own selves. We are trying to understand the cognitive functions, the brain functions. This is another great area which we are trying to understand. So the key is the world is going to move towards understanding the microbes within us, around us, and on us to come to individualized and personalized ones. My favorite area is obviously nutrition and then therapeutics. These are the two companies that I run, one in the US and one in India. The nutrition company is the one that I run out of India. And I'm extremely delighted at the way we are progressing with the launch of some of our products, which has been sort of very intelligently brought together with the help of a plant-based engine. Now, what we do basically is that we try to see that there is enough amount of diversity that is given in nutrition. And that is the key to the health of some of us. If my memory serves right, you need to have at least 52 different types of fruits and vegetables that you need to consume in a week, which I think personally none of us can do it. And so if you need to do it, we don't have to go through the age-old concept of swallowing pills and eating these what you call tablets and supplements. We can go to very good preparations or formulations which are food-based, so that nutrition can move to the next level. I mean, these are all what we call as nutri-mixes, mixes where there's a very, very careful selection of fibers that are needed for the microbes which are living within us, and so on and so forth. So this is an area that is very dear to me, and I'm sort of launching quite a number of products here, and I'm sure the world over there are a lot of people who are into this. The second part is the most interesting and challenging part, which is on the therapeutics part. I'm not into diagnostics, so I'm trying to sort of veer away from giving you any gyan on that, but I'm quite okay with discussing a few on therapeutics. So therapeutic segment is something which is amazing, because these microbes are part of us, they live within us. And so the whole idea is, can we in any way try to make a system wherein we will be able to put all these microbes together and ensure that we are able to deliver some sort of a drug or possibly an antibody, or possibly something that can disrupt an immune system, and so on. So it's almost like a live microbial cell factory inside the human being, which can be controlled to release a drug, or controlled to release something that can be very easily then done with the help of genetic manipulation. So synthetic biology will have to go one notch up in terms of putting engineering a big pathway, or bioengineering a molecule that will go inside a bug, put the regulatory switches, the safety switches, the ability to control them, and then deliver drugs exactly at the target. Now, this is very useful for diseases like cancer, where the collateral damage of running the drug through the systemic route causes serious problems. So it is a lot more easier to have them directed at a place, so that the quality of life of people completely improves. Cancer treatment is sometimes not the most human-friendly type of treatment, because basically it involves killing something in us, which is our own cell, which has turned rogue. So when we try to chase down the rogue, you also chase down some of the good things in yourself. And that is what is the collateral damage that normally happens in cancer, which is far more debilitating than actually what you might be suffering on account of cancer. So these are some of the areas that will be there. And imagine the area of diagnostics, even though I'm not going to go into great detail, but let's say these bugs go there and in advance, let you know that, yeah, something is impending, something is coming. You're going to be diabetic, you're going to be having a stroke, you're going to have something here. So these are signals that can come out, which can be measured on account of the diagnostics, where you can use the microbes as one of the tools. We have to also be very careful, in the enthusiasm to take up research, to do research, to develop an area, to bring it to the forefront, to expose it, we have to be extremely clear that, suddenly we shouldn't go the way and say, oh yeah, you know what, the next way of treating everything, the whole world is going to move towards microbiome area, everything is going to be done that way. Let's spend our time or money on this more. I mean, that's not how it works. I mean, this is another way of tackling a disease, another way of doing a diagnostic, another way of improving nutrition, another way of trying to recognize that there are a few trillion cells that can be beneficial to us. So we should not be going overboard on hyping, because that's something which we have to be very careful about. But we have to recognize that these cells coexist, and they are something that we can manipulate for our general health. So the major thrust all the while has been on, gut microbiome. I mean, there are several millions or trillions living there. So we believe that, yes, that's the way to go about it. But as you know, there are several other areas where microbes are there, which can cause problems and which can also alleviate a problem. That includes in areas like the urogenital places, respiratory system, that I'm working. Till a long while back, it was believed that respiratory system is completely sterile. It does not have any bug in it. But that's far from true now. So that's the basis of how we founded Eli Biotherapeutics in USA, where we are trying to engineer a bug to deliver an anti-cancer molecule into the respiratory system. So what's going to happen is, in the years to come, these microbes are definitely going to shape the way in which we eat, the way in which we sort of treat, and the way in which we are going to have diagnostic tools inbuilt into us. So these are all sort of live circuits that are going to communicate and interact with us, which is very good, because it's almost like you put something in to have a control over, to tell you what's happening outside. Otherwise, these guys are remaining very silent. I mean, they've always been secreting some chemical to communicate to us, but we have not. So it's almost like looking for the alien outside. Here we are looking for the alien inside, which is not an alien actually, but within ourselves. So this is the area, and this is how I think it's going to move about. Also, it goes to show that, our system, our DNA is also made up of several bits and pieces of the viruses and the bacteria and the microbes and the parasites and all that. So we are a complex amalgamation, aggregation of genetic material from different type of human and microbial species. And I think this is something Manoj will be definitely touching upon, because this is something which he knows and he's an authority in. So this is in my way, the microbiome research is going to move away from that and look at other areas as well, where there's going to be a lot of things that can be done. Now, having said that, the major challenges that come to at least to my mind, is that, at least in India and maybe in other parts, is that you have to remember, if we need to do trials for microbiome, so it's not going to be the usual 30, 40 people. You need really big clinical trials. The sizes will have to be big. Now, I don't know how you can do that, because with the current costs, with the current type of investigational methodology, how can you do trials which involves a few thousand people, unless you are a big corporation or a big pharma company or something like that. So I think there has to be some methodology, which I don't know today what it would be, but for convincing proof, we need to do a very large number of trials. And I don't know how that can be done, but the costs will have to be taken into consideration there. Now, having said that, what would happen is, if you're going to have big data coming out of it, right? I mean, if you're going to do clinical trials, you're going to have lots and lots of data coming out. Lots and lots of data coming out. We need to have a tremendous data crunching capability, which is called the big data. And the analytics part, annotating part, the understanding part, the relating part, all needs tremendous amount of work that needs to be done. So I'm looking at a research cohort evolving around the globe, which will be spending a lot of time in synergy, understanding what's within us and how can we manipulate, use it to our advantage, and to understand how we can be a much more healthier living system than we are today. So those are some of the things that we will be keeping in mind. So uh, I hope I have been able to give you as I promised in the beginning of this talk, a 35,000-foot overview of how this space is going to go into the future and how exciting this space is. And if that is the case, how can we actually exploit it to our own good, and see how we can take it to the next level. So I once again wish to thank the organizers for the excellent arrangement and the amount of advertisement that they had done all over. And I wish if there are more such seminars in the future. And if there are any questions after this, I'll be very, very happy to take them and try to answer wherever I go. Thank you very much. Arrangement and the amount of advertisement.
Okay, so that was Dr. PM Murali. So our next speaker is running late. Life of a CEO, being myself a CEO, I know that it's tough. So maybe he will join us shortly. In the meanwhile, we have Dr. Anindya available from Unilever. So he'll be joining us shortly. I will pass on the controls to him. And after that, we'll try to connect to Dr. Manoj. Okay? So stay tuned, guys, while I connect you to Dr. Anindya.
Hi, good evening, everyone. My name is Anindya Dasgupta. I am a science and technology platform workstream leader in Unilever R&D based out of Bangalore. I would really like to thank the organizers for giving the opportunity to speak to you today. And what I will be talking to you about is the host microbe crosstalk on skin, how we have developed insights and then gone on to make some applications based on those insights towards improvement of microbiome on skin and therefore the host as well. So moving on to the second slide. So Unilever has an ambition, a big, bold ambition in the area of microbiomics. And what we see microbiome and humans is as a joint ecosystem. No longer do we see microbes as them and hosts as us only or me myself, but it's a combination of both which leads to us. And the ultimate ambition is to have a balanced microbiome. And what will it do by having a balanced microbiome? It will inherently deliver a benefit to the host or the consumer. And it can be a host on face, skin of face, or hands, or scalp, feet, underarms, or different parts of the skin body. And as you know, Unilever has multiple skin care and skin cleansing brands such as Dove, Vaseline, Lifebuoy, Pond's, Clear, etc. Some of them are in India and some of them are across the globe. And we tend to cater the science of microbiome to all of our brands. So I mentioned about a balanced microbiome. Now the question is, what is a balanced microbiome? How do you define a balanced microbiome? And the answer is not as easy as you think, because microbiome among individuals vary by a whole lot. So when you say a balanced microbiome, what does that mean? Is it balanced for you, me, our neighbor? To answer that, we have come up with 5 very, very simple principles which would be a guidance to a balanced microbiome. And what we call them are the five signs of microbiome. So I'll start from the right to the left, actually. Uh, so the fifth sign of a balanced microbiome is the absence of known pathogens on your skin. Right? And today, more than ever, the presence of a pathogen on skin or a part of your body, we know how detrimental it is. So, uh, so that's number five. Number four is presence of principal commensals on your skin. These are your inherent good bacteria which reside and cohabitat and maintain a cohabitance with your skin. So those should absolutely be present on your skin. Third is the diversity of the skin microbiome. So it's not good enough to have just one or two of the commensal bacteria on skin, but it should have an optimal diversity. Various studies have identified anywhere between 200 and 400 species of skin microbes being present on a healthy skin, and they maintain an optimal diversity for their ecosystem as well as the benefit of the skin. And we should be very careful in maintaining this diversity. Number two is resilience. So every time you use a product, there is a possibility that your skin microbiome gets altered. Now, a healthy microbiome or a balanced microbiome is one that even if it's altered for the short term, it does get back to its original state, which shows the resiliency of the microbiome. That's very good. Now, we come to the first one, which is ecosystem services. What do we mean by ecosystem services? Ecosystem services actually means, yes, the microbes are there, they're living in diversity, in resilient conditions, but what does that mean to the host? Because ultimately, a microbiome is nothing without a host. So, what kind of services does it give to the host? For example, does it help the skin to get better hydrated, moisturized? So these five, and when one observes on skin microbiome, is what we call the five signs of the healthy microbiome, which gets us to getting balance. And we in Unilever have moved the needle a little bit from starting to carpet bombing, with non-specific antimicrobials towards targeted kill, towards giving benefits without kill but mitigating the bad effects. We've also moved into the area of preserving the good in the last few years, and you'll see in a few examples. Uh, and then nurturing the good bacteria, and then finally, your body knows best. So, how can the bugs and your body's innate defense system work together towards benefits? And we've not done this alone. We couldn't have. Over the years, we have built many collaborations across top universities in the world, from the far east all the way to United States, and with India and Asia, other parts of Asia and Europe in between. Uh, we have had 16 years of R&D in this area. We've published more than 100 papers or formal presentations in conferences. We've filed more than 75 patents in the area of microbiome and antimicrobials, etc. And we also work with a number of partners to win suppliers who all form along with us our own integrated ecosystem, if you may. And besides the science, we are a commercial organization, so we make products towards the benefit of consumers. And as you can see, this illustrates the number of launches which are in the area of microbiome, be it microbiome gentle for Dove, or be it prebiotics for baby Dove, be it anti-acne, which balances the microbiome of face through ponds, through then working with skin's immunity through Lifebuoy, ending dandruff by again balancing the microbiome by killing of specifically of the Malassezia causing fungi, etc. So let's get back to the science a little bit again. So the skin and microbiome, I said it's a joint ecosystem. What do we mean? They are in constant interplay. You cannot separate from the others. For example, the commensal bacteria actually secretes bacteriocins and short-chain fatty acids that inhibits pathogens. On the other hand, these bacteria also make things which are beneficial for the skin, like it makes lactic acid and free fatty acids. The skin, in turn, has also developed mechanisms by which it controls pathogens and help in growth of commensals through its acidic pH, through hydration, and through the natural system. So, as you can see, it's a very complex, but in peace at most of the times, ecosystem. And the bacteria on skin, we like to call them, in this way, we have the good, bad, and the ugly. So good or and bad are quite clear. The ugly ones are something like a staph aureus, which are not necessarily bad for the skin, but in the underarm they are the main producers of thiol-derived malodor. So you want to, if not kill them, at least you want to stop the malodor in the underarm. And here is an example in the next slide where you see, this complex interplay in place, in a dysbiotic condition. So this is an example of atopic dermatitis where, there is dry skin and inflammation and itching, etc. And then you see there is a host element to it in which, the water evaporates very fast from the skin. There is inflammation and a lot of inflammatory markers come up. Epidermal barrier disruption happens. Filaggrin synthesis goes down, etc. So this is what happens to the host. Now, what happens to the microbe is also very, very interesting. If you look at the signature of the microbiome diversity in atopic dermatitis, you see a massive difference with control, as you can see as the colors indicate. And if you look at the numbers, the most interesting part is this staph aureus. From almost very negligible amount of staph aureus from healthy skin, the mean relative abundance goes up to about 21%. Con- Conversely, Staphylococcus hominis, which is present in a decent amount in skin, normal skin goes down. Again, an example why we call it something like a hominis, an ugly bug, but not a bad bug. And this is all derived from the alpha diversity index. So as you saw in the previous slide, there's a change in host, and concurrently there's a change in microbe. So it's very, very clear now from scientific evidence, and this is not just our data, data from others as well. There's a very complex interplay between the host microbiome and the host. I mentioned about pH, right? And here is one set of experiment where we did to show that how critical the pH of the skin is. Now, what was done in this experiment is we grew two bugs, one which is a good bug, or epidermidis, and the other an ugly one. And we looked at the growth of pH growth of the bug as a function of. What was very clear in S. epidermidis was that when you grew bugs at a pH of 5 versus a pH of 7, there was a small but almost negligible difference, right? Whereas if you now grow S. aureus, which is a pathogen, what you see is a remarkable difference, that they are quite incapable of growing under acidic condition, but once you put them under neutral condition, they grow like gangbusters. So this is the pH 7. Now, how does it relate back to skin? Remember I said that in atopic dermatitis, the skin pH is around 7 almost, or 6.5, whereas normal skin pH is around 5.5, right? And so the skin pH does, in atopic dermatitis, actually facilitates the growth of a bad bacteria like S. aureus, whereas the acidic pH of a healthy skin facilitates the growth of a good bacteria like Acinetobacter. And what you see in this model and this growth curve is exactly the meeting point, which is, as you say, see again, SRES grows very well under alkaline pH, whereas skin commensals like SEPI, there's not much of a growth. In fact, if anything, for hominis, it actually drops. So again, this shows a very nice correlation between the acidic pH of skin and skin health and the growth of good bacteria, whereas disturbance of the acidic mantle of the skin and driving it towards a neutral pH actually causes growth of bad bacteria like SREs. Having spoken about the general host microbe interaction and the effect of host on microbe and vice versa, let's move on to some areas where you can actually intervene. And all of you are quite familiar with these terms: probiotics, prebiotics, and postbiotics, where probiotics consist of addition of live good bacteria to products. Prebiotics is adding good food to the good bacteria on the skin. And postbiotics is when good bacteria uses prebiotics and makes a fermented product. Those are called probiotics. So in Unilever, we've taken the approach of prebiotics as a technology route towards providing benefits to consumers. And we defined probiotic as being utilized by good bugs to produce various metabolites, and these metabolites then go on to either inhibit bad bacteria or promote skin benefits. And there are many ways we measured this. We measured the growth of S. epidermidis, which is a known commensal bacteria. By prebiotics, we see the utilization of the nutrient, and thereby, if it grows well, it's a prebiotic. And then looking at the ferments, if it inhibits harmful bacteria and generates some good things for the skin, it fulfills the criteria of inhibiting bad bacteria and skin benefits and providing skin benefits. So, to cut a very long story short, when we fed cells with prebiotics, fed SAP with prebiotics, we saw each of these three things happening. First of all, we saw that a prebiotic actually helps in growth of S. epidermidis significantly by about 1.5 log. Uh, However, a pathogen like S. aureus was not able to use this prebiotic, so which is good. So it selectively enhances growth of good bacteria, but doesn't do anything to the bad bacteria. And going back to inhibition of bad bacteria, if you look at when we took the prebiotic ferment and allowed growth of uh, S. epidermidis, again, which is a commensal bacteria, and S. aureus, which is a pathogen, you see that that ferment did not do anything with the good bacteria, but it selectively inhibited growth of bad bacteria. So again, that's a very, very nice. So, and the most exciting thing that we saw was when we did um, a analysis of all the metabolites that are produced by the prebiotic bacteria, with the prebiotic agent in the present by S. epidermidis, it led to production of several things, and most of these are actually short chain fatty acid and lactic acid. Lactic acid is a known skin moisturizer, anti-aging agent, a resilience marker, etc. It's got many, many good benefits. And we looked at it numerically, we saw almost like a 5 to 10 fold increase of lactic acid, when S. epidermidis used it. And this was an in vitro assay when we took actually pure prebiotic and added it on skin, and 72 hours later we extracted using a buffer system. We saw about a threefold increase in lactic acid on skin by the use of prebiotics. So that's all good. So it helps good bacteria grow specifically. The ferment specifically help the bad bacteria to be controlled, and then it helps in skin and skin bacteria making several goodies which are beneficial for them. So that is one example where we've been able to use a prebiotic, and in this case, it's as a simple one as glycerol. So in conclusion, we do have a check and balance system in the skin microbiome, where the SAP, the skin pH, they all are almost like walls together, as is the bad bacteria and the rise of skin pH. Okay? I'm giving you the example of atopic. And then, of course, there will be always fluctuations. There will be product usage, environmental insults, etc. But if you go back to the 5 signs, remember the resilience. So a balanced microbiome would always be resilient. And even after a harsh product use or environmental insult, it can bounce back to its original. So I'm at end of time now. I would like to thank all the people who have contributed in this. In Unilever, we have 5 global labs, and we work very, very closely together. I want to thank all of them. And last but not the least, I would really like to thank the organizers to give me this opportunity to come and speak to you. Thank you very much.
So that was Dr. Anindya. 58 minutes into the webinar, 2 speakers done. How do you feel? Everybody, quickly in chat. Eager for the next speaker? Okay, great. So our next speaker has arrived. I know CEOs are tough to catch, and Mr. Manoj is right here. We dialed up to USA to make sure that he is right here with us. So I would add him to the screen now. Yeah. Hi, Manoj.
Hey, how are you?
So, doing great. How are you?
Doing great. Sorry for the panic.
No problem. So, the dice is all yours. You need to share your screen and get started.
Believe it's shared. Can you hear me okay?
Yes, we can hear you. Perfect. Please go ahead.
All right. Thank you, everybody. Thank you for the organizers for the invitation. Very much appreciate it. My name is Manoj Dadlani. I'm the CEO of Cosmos ID. And today I'm just going to give you an overview on how we're approaching different areas in the microbiome through combination of sequencing and bioinformatics, and also what we're thinking about for the future in the microbiome space. So thank you again. Just a little bit of a background on Cosmos ID. What we've done is we've built a platform to enable microbiome product development. And we focus in 4 different areas. The core of everything we do is on the bioinformatics side, so that really leads to the high value add interpretation from the microbiome sequence data, and we apply the bioinformatics tools and software that we've built in a few different areas. One area is contract research, so where we do work with pharma companies and consumer companies in their product development and their R&D. We also work with direct-to-consumer companies that are doing direct-to-consumer testing. And then finally, the other side, obviously, there's good microorganisms, and there's also pathogenic microorganisms, as we're all deeply aware of today. And so we also do work in the infectious disease diagnostic space, utilizing sequence data and these bioinformatics tools that we built. So as a company, we focused in a pretty broad set of categories. So as I mentioned, pharma, both on the therapeutic side, the clinical side in terms of diagnostics and epidemiology, nutrition, primarily probiotics and wellness areas in terms of nutrition there, the ag space, consumer testing. So skin microbiome is pretty big area for us, and I think we just saw some great data in that space. And then applied testing. A lot of great partners: big pharma, small pharma, government agencies, universities, companies, and then we also partner with the data generation companies. So Illumina, Thermo Fisher, PacBio. We're doing some more work with Nanopore now. So pretty broad, in the sense that microorganisms are everywhere. We kind of touched on a lot of different areas through our work. So I'm going to run through some examples of different areas we've worked in. I'll start with pharma. And so when we think about the pharma and the consumer product R&D pipeline, we really think about it in a few different sectors. The first side of it is the target discovery side, and your early research, where you're trying to understand what your targets are, how those can improve health, what are the functions that you're targeting with those targets. And then once you have identified targets, you obviously have to go through regulatory approval, so you're looking at safety and efficacy. And then once you've confirmed that your product is safe, you obviously have to scale it, produce it. And since these are live organisms, many of the times you have to control that those organisms are not mutating and not changing, or acquiring genes that would not be beneficial. So it's a big end-to-end view on how we approach these product development projects. So an example here is with a company called Synthetic Biologics. They've developed a drug called Sino4, and it's basically a drug you take along with a strong antibiotic. And the goal of that drug is to reduce dysbiosis in the gut to prevent secondary infections. And what you see is we did a trial with them where we looked at 3 different cohorts. One cohort is just your baseline. So what does the gut look like before taking any antibiotics? And the next cohort is when you take an antibiotic, a strong antibiotic like ceftriaxone, you can see this huge amount of dysbiosis in the gut versus the baseline. But if you take their drug along with the strong antibiotic, you see that the dysbiosis is quite minimal. And what's actually really interesting is that there's a specific organism, Methanobrevibacter smithii, that you see an overgrowth in when you take ceftriaxone by itself. But when you take ceftriaxone with Sino4, you don't see that overgrowth. And this bug is actually known to lead to secondary infections such as C. diff, which is one of the main things that Sino4 is trying to prevent. So through this high resolution microbiome analysis, what we're able to see is that this drug does have efficacy in this case, and they were able to move on further in their clinical trials. Go through another example in the consumer product space. This is a really cool product that we're working with. It's a company called Symbiome. They're based in San Francisco, and they've really focused on bringing the ancestral microbiome back to present day, you could say. And they're actually working with a tribe called the Yanomami tribe in South America, and it's basically an untouched tribe where they've been working with them to understand their microbiome and see what we've lost through industrialization and all of the different things that have happened over the years, where our microbiomes have become less natural and less grassroots. And what they're trying to do is understand what those differences are and bring them back in products to restore health and restore beauty in the skin. So this is their first product, which is launching very soon. But the amount of research that they're doing is actually really exciting. So we've done some very deep shotgun sequencing of members of the Yanomami tribe. But there's also a really cool story behind this. There's a man named David Good, who is an original Yanomami, and he came back to the U.S. And grew up in the U.S. And his mom stayed back in the Yanomami tribe in South America. So we were actually working with David, who is a biologist, and he actually went back to the Yanomami tribe and spent 3 or 4 months there. And we sampled him before going to the Yanomami tribe and when he came back to see what he picked up. as well as we sample the Yanomami tribe to see how they're different than a traditional healthy or quote unquote healthy American, to understand what we've potentially lost through that, through the industrialization. And so we didn't only look at what those organisms are, but we've also looked at a lot of different functional aspects of these profiles. So things like purine metabolism, biotin biosynthesis, DNA recombination, quorum sensing, and a few other functions, where we've really dove deep both into who is there from a taxonomic standpoint, but as well as what are they doing from a functional standpoint. And Symbiome has been a great partner because they're very keen on very in-depth sequencing and in-depth research. So we're using full shotgun sequencing, very high read depth. We're doing both DNA and RNA sequencing. So we're looking at the metatranscriptomic side of things as well. And down the road, we're also looking at metabolomics and trying to understand both from the metabolite standpoint and matching it to the genomic side, how these differences come about, and how we can possibly restore that original beauty and glowing skin back to today's consumers. Another area that we work in is the probiotic space. So here, one of the things is really just understanding is what is in a probiotic actually in the probiotic. So this is some work we did with the US FDA, where we looked at different probiotics to understand are the strains and the quantities of those probiotics accurately represented on the label. So here, what you see is 4 different probiotics: probiotic A, B, C, and D, where the organisms in blue are what's expected to be in the sample that's on the label. And what you see is in B, C, and D, they pretty much align well to what's expected. In probiotic A, you've got a few different potential contaminations in that probiotic. So really using metagenomic sequencing to understand, are the right strains in my product? Are the right quantities in the product, as well as is there any potential contamination in the product? And that helps really ensure that we're generating the right probiotics, as well as making sure that they're safe. Taking that a step further is really how deep can we get into these individual probiotics and their organisms. So here we can really get down into understanding SNP level differences between different strains. And so with some companies that we work with, we actually take each of the individual organisms in their probiotics, and we sequence them whole genome sequencing to understand how similar they are to what they expect to see in the sample, as well as is there something novel in those types of probiotics. And you can do that with whole genome sequencing of different isolates and really get to very high level SNP level differences of the isolates and of the strains that are going into your probiotic. And then from a safety standpoint, looking at different types of genes that could be harmful to individuals. So another safety screening that we do with our probiotics customers is there antimicrobial resistance genes in the individual strains or the mix of strains that they have in their probiotic. So obviously you don't want to increase the antimicrobial resistance burden in your consumers by giving them probiotics that have additional antimicrobial resistance genes. And so this is one additional safety screen that also goes along with virulence factors. And virulence factors are genes that lead to pathogenicity. So a screen of different probiotics for these types of genes is also really important to ensure the safety of the product that's being released, as well as to ensure that your new batch of strains or product is not acquiring antimicrobial resistance genes or virulence factors over time. Whereas it could be safe today, but in your next batch tomorrow, if they've acquired some sort of genes, you want to make sure that isn't the case before you release that product. So another area that we work in is gut testing. And this is going to kind of the growing direct-to-consumer space that we're seeing in the US, and we're also seeing it in Europe, and actually have had some discussions in India as well on how you can do gut testing to evaluate and monitor health. So one of the products that we launched this year is called BiomeFX, and we launched this with a probiotics company called Microbiome Labs in the US. And what's unique about their approach that we worked on with them is that they had a couple rules that they wanted in developing this product. The first one is that it couldn't be 16S based. It had to be based on whole genome sequencing. The reason is with whole genome sequencing, you're able to get much more information than you can get with 16S. So with 16S, you're able to get basically lower resolution taxonomic profiling of bacteria. So really just to the genus level. Whereas with whole genome sequencing, you can actually get down to the strain level for bacteria, but also look at viruses, fungi, protists. And similarly, you can look at antimicrobial resistance genes, virulence factors, and you can have much accurate or much more accurate functional prediction, which is why they called it BiomeFX, because they really wanted to focus on the functional side of the gut microbiome. We also worked with them to develop a more representative stool sampling method, so you can get more representation across the entire stool as opposed to just one area of the stool, because we've seen differences in profiling at different parts of the stool. And then we worked with them to really come up with a nice report that has recommendations, again geared towards what's the function in the gut. So here's some snapshots of the report. There's actually a version 2.0 coming out next month, which adds new functional analysis and recommendations in that report. So and you can find more information on the BiomeFX website. The last area that I had mentioned was infectious disease diagnostics. So I think we're all deeply entrenched in COVID-19 today, and really some of the rapid advances in COVID-19 testing came from rapid sequencing in China of the Wuhan strain. But outside of that, infectious disease diagnostics is obviously a very important area, but also one that leverages the same sequencing and bioinformatics technologies that are available in the microbiome space and that we're focused on at Cosmos ID. Well, one of the maybe scarier statistics is this is a report that came out of the Wellcome Trust in the UK, but the track or the projection on superbugs leading to death by 2050, the prediction is that there'll be more deaths from superbugs by 2050 than there are deaths from cancer today. And that's pretty alarming in terms of statistics. And if we don't really improve how we monitor and diagnose infectious disease, as well as how we are more tailored in our treatment recommendations for antimicrobials, we'll keep increasing that resistance, which will lead us to have less available drugs and more deaths due to these superbugs. So we've developed a software platform for infectious disease diagnostics called Cosmos DX. It's a cloud-based platform. It's HIPAA compliant, and what it does is allows you to analyze sequence data and give very accurate results for what organisms are in that sample, as well as antimicrobial resistance genes, the affected drug classes, and the generic drugs that are affected in those samples. And what it does is, as opposed to kind of the research side of things where you're looking for everything and anything under the sun, with this platform, it really leads to a clinical report that can be used by a doctor to eventually diagnose and treat a patient with an infection. So this is kind of another flavor of bioinformatics and microbiome sequencing for the infectious disease diagnostics application. So a little bit more on Cosmos ID. We have two sides to it. Obviously, one is very entrenched in the bioinformatics side, and we've built the largest curated databases for bacteria, virus, fungi, protists, antimicrobial resistance, virulence factors. And what we've known for is very accurate strain level identification. The other side of it is we have an NGS service lab that's CLIA certified and GLP, where we do end-to-end sequencing as well. So shotgun, amplicon sequencing, as well as isolate sequencing. I keep mentioning strain because strain is really the clinically informative and actionable unit. So going back to your biology days, strain is kind of the highest level of resolution in the taxonomy. And what we've been able to do is give very accurate strain level resolution. And you might ask, why is strain level resolution important? So here's an example. Lactobacillus casei species. one strain of that species could be used to make cheese. Another strain could be used in producing Chardonnay, and a third strain could be used in a probiotic, like a yogurt. All of those are great outcomes of these three different strains, and nothing really is alarming or concerning. But if you take another species like E. coli, you could have commensal E. coli that are beneficial and support in things like vitamin production, host protection, immune support, digestion. It even protects against some pathogens. But then you could have a strain like O157H7, which is highly pathogenic and cause hemorrhagic enteritis, which could make you very sick or possibly even die. And really being able to differentiate between these different strains is important because they infer different functions and potentially different effects on the health of humans. So again, we work with researchers, organizations, end to end, study design, sample collection, sequencing, bioinformatics, in-depth interpretation. We also support in sample collection and kitting. In cases where you're looking for a clinical trial or multi-center clinical trial, we can support there. Also with direct-to-consumer testing, we can support the end-to-end process. And I think what's key is the bioinformatics is really important, but it's really a sense of garbage in, garbage out in general. And if you are not producing high quality data as your input data, it's really going to limit your bioinformatics analysis at the tail end. So we've worked a lot in standardization of the workflow to minimize bias, and that covers what type of controls we include, a lot of the testing we do in different extraction methods for different sample types, different library prep methods, ensuring that we reduce or prevent carryover and crossover contamination. And these are all different aspects of the wet lab process that can really introduce bias. And then on the dry lab or the bioinformatics process side of things is another area where we've done a lot of innovation in terms of both database curation, filling in holes in databases, improving the balance between sensitivity and selectivity, reducing false positives, and being able to give very high resolution classification down again to the strain level. So as I mentioned controls earlier, we also adhere to a lot of different laboratory standards. So GLP is a rigid standard. We're also CLIA certified. So we have routine inspections from the government there on our certifications. We're also HIPAA compliant. And I think by having these rigid standardized methods, as well as adhering to these different standards, really ensures that we produce high quality data, which we can then have the best results in from our bioinformatics. And again, I mean, price has always been a challenge in the microbiome field. We've done a lot of innovation and a lot of scaling to really bring down the price of sequencing of microbiome samples. So we're able to do whole genome sequencing starting at $99 a sample, and that includes the bioinformatics results down to the strain level. And what we've done is to make this, to get to this price point, we've really looked at a few different things. So one important thing when you are deciding how deep you sequence a sample is a few different considerations. And the goal when you're sequencing is to really get as much microbial representation as possible. And things that can affect the microbial representation are one is how much or how many microbes you have. So if you have many microbes, you need to sequence deeper in order to get the characterization of that entire microbiome profile. And the other major one is how much host DNA you might have in the sample. So an example is in stool samples, you have higher diversity, but you have very low amount of human reads in that sample, which allows you to sequence at a lower depth in order to get to the microbial, a good microbial representation. However, with something like saliva, where you have a very high amount of diversity, but you also have a high amount of human reads, in order to get those human reads, you really have to sequence very deep to capture those reads from the sequence data and have a representative enough data to analyze and make accurate predictions on. So for us, it's been really around understanding the different sample types we work with in order to recommend the right read depths in order to get an accurate representation from the bioinformatics. So finally, just again, diving into the bioinformatics more, there's a few major components that go into the accuracy of your bioinformatics. One part is the database. And in the public domain, there's a lot of misnomers and misrepresentations in what goes into those public databases. So what we've done is we've really spent a lot of time and energy curating public databases, as well as adding in data ourselves from collaborations we've had and actually organisms we've sequenced on our own as well. So we've built the largest curated databases of 160,000 genomes and gene sequences for commensal organisms, pathogenic organisms, and environmental microbes. And then what we've done is we've organized this database phylogenetically. So you can think of this as a like evolutionary tree of life, where we can mine those that database down to the strain level, and using the algorithms we've developed, give very accurate strain level resolution. And what we've done there is we've layered on machine learning to improve the sensitivity and precision by analyzing tens of thousands of samples that are very different. So human samples, animal samples, environmental samples, and the mix of all other types of samples to really improve that false negative and false positive rate. And then we make it very easy. So anyone's welcome to sign up. If you just go to app.cosmosid.com, you can actually set up an account and play around with the bioinformatics and example data sets in the application. And it's literally point and click. So you don't need to know how to code, and you're able to give very quick and accurate primary analysis. So that's both down to the table and statistics in terms of organisms, but as well as comparing samples to each other in different cohorts and looking at different visualizations, all with pointing and clicking. So our goal is really to keep it as simple that our accountant who knows no bioinformatics can actually go in and actually do analysis on our platform. And that's kind of our test case. And then the next part of it is really looking at the functional side. So as I mentioned earlier, is metabolic pathways, molecular function, cellular components, biological processes. So it's not just who is there, but it's also what they do. So for example, you could have kind of a disease cohort, which then you treat, and you can see how that treatment group compares to a baseline healthy group. And you can see how different functions are missing or restored through some sort of intervention. But really, the function is kind of the future in the microbiome space, and understanding how to analyze and interpret that data is what's kind of our newer focus and what's really coming next. So just wrapping up in terms of accuracy, this is some internal benchmarking. This is a study that came out of Weill Cornell. When we've taken those same data sets and analyzed them and compared them at the species level, our species level resolution was better than all of the tools that they use, many of which you might be familiar with, such as Kraken and Clark and Metaflan. You can see where we really shine is at the subspecies or strain level. The F1 score is the harmonic mean between precision and recall. And this is where you see our performance stays really high, but all the other tools really drop off. And this really comes back to how we've built our databases, how we've curated our databases, and the algorithms we've built coupled with the machine learning to improve the accuracy. So internal benchmarking is great, but if you look at kind of third-party validation, we've entered multiple community challenges for bioinformatics accuracy. And we've been honored to win all of them. So we won the Mosaic Strain Challenge, which is put on by Janssen Pharmaceuticals, and we're the #1 performer here. And we also won the Precision FDA CIFSEN Pathogen Detection Challenge for the most accurate strain level identification and the lowest false positives. And the CDRH BioThreat Challenge, which is the most recent challenge put on by the FDA. There's 29 submissions from 11 different contenders. And you could submit multiple entries, and out of the top 5, we placed first, second, third, and fifth. So a great third-party validation in terms of the performance of our bioinformatics. And again, the secret sauce is the databases, the curation of the databases, the algorithms we've built, and the machine learning we put on top of that. So where are we going? And a lot of this is focused on the bioinformatics side. What we see next is really 2020 and beyond is multi-omics data integration. So I mentioned metabolomics earlier, and really being able to look at multiple sets of data to think about the microbiome and how it fits into a systems biology standpoint. We'll continue to enhance our taxonomic and functional databases, and we're working on adding more machine learning and predictive modeling, really with the goal of going towards artificial intelligence. And the first step that we have to tackle is how do you get real big data? Because really right now in the microbiome space, the number of samples isn't really in the category of big data. It's really just starting. So things like cost optimization and helping researchers and companies generate large amounts of data for lower cost points is the first step, because then when we have big data, we can actually start mining it. And so the next step is predictive modeling. And so where we're focused on moving forward is really the exploratory data analysis from these robust data sets. So improving the statistics, improved regressions, really driving into real time series data, and then looking at other qualitative methods to improve the data input as well as the predictive modeling. And there's been a lot of development in machine learning and deep learning that can help us with this predictive modeling and predictive capabilities that we're working on developing. I think again, still for the amount of data we have today, we're really kind of in that machine learning stage. But there's a great amount of tools out there where we don't necessarily have to reinvent the wheel, but we can adapt. And both from the machine learning side as well as the neural network side, even things that are really picking up like TensorFlow and how we can apply that to microbiome data is really where we see the future and where we're investing today. That's all I have from my side. But again, you can see our publications on our website. Feel free to play around with our bioinformatics tools at app.cosmosid.com. My email is manoj@cosmosid.com if you ever have any questions. And again, thank you for your time, and thank you for the organizers for inviting me.
Thank you so much, Mr. Manoj, for your time. It was really a pleasure having you on board. I hope I'm audible.
Yeah.
Manoj? Yeah, okay. So we had planned for a Q&A session, but it's getting late here. So unfortunately, we'll have to cancel that. But nevertheless, I would like to thank you for your time. And people, I hope you enjoyed. Yes? Okay. Fantastic. So that was Mr. Manoj from Cosmos ID. And now I have a special guest who represents KTech, Government of Karnataka. Let me add him. Yeah. So the dice is all yours.
Yeah, thank you, Shekhar. Good evening to everybody. Thank you for joining on the Saturday evening. It is our pleasure to talk to you and give the the emerging area, what we think microbiome, which we identified as one of the emerging area under Karnataka biotechnology policy. So we thought it's a good time to celebrate the World Microbiome Day. At the same time, bring awareness among the biotech community, the diversity that is available in terms of the microbiome and the applications part of it. So I really thank all the speakers for bringing the perspective and the perception of that. And it has been a fantastic talk given by them. And they readily agreed when we requested them to give a talk here. They readily agreed. I thank all of them for the whatever support they have given to us, and I thank Abel who have been our support all the while, and special thanks to Mr. Shekhar Shuman and the BioTecNika team. And they have been very cooperative, and they've been a wonderful platform for us to convey the details what we want to do. So basically, we want to promote technology and more so emerging area technologies, so which is very critical for coming with the innovative ideas and support the the healthcare system what we are looking at. Keeping that point of view, I hope you all had a good insight into the microbiome. And I think many of you among you will be inspired and take this one of the studies. With that, I thank my colleagues at Biotechnology Facilitation Cell in KTech, and I also thank my managing director, who has been very supportive. With this, thank you very much, all of you, for not joining on this Saturday evening.
Okay, all right. So that was Dr. Bala, one of the facilitators, one of the coordinators who helped us a lot in conducting this mega webinar. My dear friends, today you witnessed 3 CEOs and 1 global leader of Unilever. So I know you have a lot of questions. I know you all are bubbling with a lot of questions, but you can see the time. It's already 8:52, and if we start taking up questions, it will go until 10 o'clock. And let me remind you, CEOs are busy people, so it'll be really tough for us to take up every question, and just to make sure that doesn't happen. So we won't be able to take any questions as of now, but I hope you all enjoyed this session. It was really a pleasure talking to all of you. And now I would like to thank ABLE, Association of Biosciences, Biotech-led Enterprises, KITS, KTech, and Government of Karnataka, and in particular Dr. Bala and Mr. Suresh, COO at ABLE. I would also like to thank the entire BioTecNika team who made this mega webinar possible, and especially to the online team of BioTecNika. And of course, thanks to all of you, my beloved audience. It was really a pleasure interacting with you on this Saturday evening. We look forward to interacting with you once again. And thank you so much for attending this webinar. Now is the time for the feedback form and the e-certificates for all those who want to mark their attendance. So yes, the feedback form links will be sent to you in your chat box right now. But before you leave, please make sure that you have liked and subscribed to BioTecNika. We will keep coming up with more such international audiences, CEOs and managing directors, and various industry stalwarts who have created the foundations of biopharma industry globally. But that cannot happen if you do not subscribe to BioTecNika and like this video. Do not forget to share this webinar link with all your colleagues and let them know how fantastic this webinar was. I can see a lot of fantastic words coming in the chat section. Thank you so much, and wish you good luck in your life, in your career. Stay safe, and we'll see you soon with our next webinar. Thank you once again, everybody, KTech, Able, and all my beloved audience. Bye-bye. Good night.